Greetings, class, and welcome to Chapter 9, Patterns of Inheritance. In this particular chapter, we're going to be looking at genetics. We're going to be looking at Mendel's laws. We're going to be looking at some variations in special cases on Mendel's law and some non-Mendelian genetics. We're going to look at the chromosomal basis of inheritance, and then finally we're going to close up by looking at the sex chromosomes and the sex-linked genes that can be found on them. Let's start first by beginning the unit by defining what the word heredity is. Heredity is the transmission of traits from one generation to the next, passing on of, traits from parent to offspring and offspring on to further offspring. Now, the people that study this field of science, they're called geneticists, and this branch of science is called genetics. Many consider Gregor Mendel the father of modern genetics. Now, when we're talking about genetics, we're looking at things like character. Characters are heritable features that vary amongst individuals. And the different forms, the different variations, we call those traits. And those variations can be passed on from generation to generation. Well, Mendel hypothesized whether there was different alternative versions of the traits, or genes as we call them today, what he referred to as factors back then. The different forms of the gene are called alleles. And these are the units that determine the heritable traits, what actually gets passed on from parent to offspring. Here's a picture of Gregor Mendel right here. He was an Augustinian monk, and it's kind of hard to imagine that his work was basically ignored for generations. His work was done in the middle part of the 1800s, but it wasn't until the early part of the next century, moving into the 1900s, that his work was actually rediscovered, because many of the people at the time were more consumed with the work that Darwin was doing. But ironically, Mendel's work actually explained a lot of what Darwin was talking about. And what did Mendel do? Mendel worked with the humble pea plant. The pea plant was a perfect organism to create what's called a model system. And a model system is something that you use to study when you can't really easily study what you actually want to study. So to do genetics on humans would be very complicated. But you can study patterns of genetic transmission of information and stuff like that through the pea plant and other organisms that are similar, and extrapolate that to what you would expect to happen in humans. And so he used the pea plants very simply. I think I kind of said he did it for the heck of it, but he chose the pea plant because it was easy to work with. It's very easy to make reproduce. They can reproduce in large quantities very quickly. There's only a small amount of traits, as I'll show you in the next slide coming up, and they're very easily to discern. And there's really no ethical issues working with the pea plant. So it makes a perfect model system. And basically what he did is he controlled the process of pollination, which is how plants reproduce sexually. He would remove the male part of the flower, and then he would actually take the pollen and he could transfer it to whatever particular plants that he wanted to make the crosses. And then he would observe the offspring. So he would get a lot of offspring in a very short amount of time, so he could get incredible amounts of data. And these are some of the traits that are available in the pea plant. So you can see there's not a lot of them, but they are very distinct, so you can easily discern between one or the other. Now, from his work with the humble pea plant, Mendel came up with some different laws that we still recognize and acknowledge to today. And so these are some hypotheses that he came up with. First, there are different versions of genes, or alternate versions of genes, and we call those alleles, that account for the variations in inherited characters, the thing that we see in the offspring. For each character in an organism inherits two alleles for a gene, one from each parent, one from the paternal side, one from the maternal side. An organism that has two identical alleles for a gene is said to be homozygous, or sometimes we describe them as purebred, or true breeding. If they're heterozygous, we say they are hybrid, which is a mixture of the alleles, so they have one of each kind. If two alleles of an inherited pair differ, then one determines the organism's appearance, like the physically observable appearance, and we call that the dominant allele. And the other has no noticeable effect on the organism. It's masked by the dominant, we call that the recessive. And a sperm or egg cell can only carry one of the alleles. Remember that from meiosis, the diploid cell becomes haploid. It brings on half the information, and then the other half comes from the other parent during fertilization. And so the separating of the alleles during that process of meiosis is called the law of segregation. So when Mendel did his work, basically what he would always start with were true breeding plants. Those were plants that he knew predictably one generation after generation would provide the same type of trait in the offspring. And so he would cross those. And when he crossed them in the P generation, which is the parent generation, in the F1 generation, or the first filial generation, he always found that all the specimens would have one of the traits or the other. So in this case here, we see the purple flower, which we see here. We don't see, like we might have predicted, a kind of blending of the colors, maybe a light purple or something, maybe pinkish. No, it was actually purple. It literally looked like one of the parent or the other. And so he coined that to be the dominant one. And then he would cross them again. So he would cross these hybrids, which were mixed. Even though you can't see it from actually looking at it, the genetic information was mixed. The alleles were mixed, so that in the F2 generation, one of the traits would reappear again that seemed to have vanished. And this was the recessive. So he kind of concluded that it wasn't that this particular trait was lost or destroyed or gone or whatever. It was hidden and masked by the dominant, but it still remains behind and could be seen again in successive generations. So these hybrid plants, we call them carriers because they carry the recessive allele. They don't exhibit it. If you were to look at the dominant version in purebred form and the hybrid version, they would both look exactly the same, but they're different at the genetic level. Their genotypes are different, not their phenotypes. And mathematically, he always found this ratio, 3 to 1. It didn't matter what trait he looked at, that F2 generation always revealed itself 75-25. Now, when we're talking about the hybrids that we just mentioned in the F1 generation, this is what their allele combination would look like, capital P, little p. That's called the genotype. The genotype is the actual allele combination, the dominant and recessive, the dominant, recessive, whatever the alleles are, and you get one of those from each parent. And we can use a Punnett square to show the possible combinations of alleles that could occur when the gametes combine. So the Punnett square allows us to see the segregation around the outside, if you can kind of visualize it, and then on the inside, where the alleles come together, that represents the combinations of gametes when fertilization takes place. So if you go back to that example I just showed you, this is the P generation, the parent generation. When he crossed them, the F1 generation, this was the hybrid generation where they were mixed, capital P, little p, whereas this was capital P, little p, little p. Okay. The F1 generation was like that. And then here are the parents, capital P, little p. And then the Punnett square, which we see here, allows us to see the possible combinations of the outcomes when fertilization takes place. So this is a possible outcome, this is a possible outcome, this is a possible outcome, this is a possible outcome. Now remember, every diploid cell has a pair of homologous chromosomes. Okay, one from your mom, one for your dad, paternal and maternal chromosome. And it's those chromosomes that carry the alleles for the genes, and they're located at the same locations. Remember, we called that the locus. Okay, but you're only going to pass on one of those chromosomes, but that's okay because the genes are going to be there. What's interesting is which one of the chromosomes gets passed on, because the gene information might be for a particular different version of that trait in terms of, like, say, for example, we're talking about the way the ear is attached. In both homologous chromosomes, there will be a set of code in the same place that will code for how the ear should be shaped or look, but one might be so that the ear is attached, one might be so the ear is free or dangling. And this is what I'm talking about here. So here's our homologous chromosomes here. And you can see they each contain the same information. One is the paternal, one is the maternal. But you can see in the same spots here, this is where information is. Okay, but through meiosis, only one of these will get passed on. Okay, they'll separate out. Remember, that's the segregation, the separating out. Which allele will get passed on will be determined by which chromosome gets passed on. So it could be the dominant or it could be the recessive. So this all led Darwin to kind of recognize that genetics is not some kind of accident. It's not like witchcraft or magic. It's probability. It's based on mathematical chance. You could predict with a certain amount of certainty, I guess, that's kind of redundant, the chance that something would occur, like ending up with a yellow pea color or a green pea color or a purple flower or a white flower. And we use the Punnett square to represent these possible different outcomes, these mathematical probabilities. The Punnett square was not devised by Charles Darwin. It was not devised by Gregor Mendel. It was devised by an English scientist by the name of Reginald Punnett later. That's why it's called a Punnett square. And if you remember from your middle school days, a Punnett square is the tool that we'll use to actually show the combinations of possible percentages of the chance occurring. Now, there's a lot of vocabulary, in case you forgot it all. It's all here for you here. This is in your presentation as well. This is all the vocabulary that we'll be using when we talk about Punnett squares. So make sure that you take a chance to review it. Okay. Most all of these we've kind of already talked about. This is homozygous dominant. That means the alleles are the same, but it's both the dominants. Homozygous recessive, heterozygous where they're mixed. All these are examples of what are called genotypes, which is the gene combination. And then the phenotype is the physically observable trait, the thing that you can actually see. Remember, recessive alleles are always indicated by a lowercase, dominant is a capital, and usually the letter will either reference the trait or something to do with the dominant allele, but you use the same letters. Don't choose different types of letters. It's the same letter with the capital and the lowercase. So how does a Punnett square work, in case you forgot? Okay, if one parent is capital A and little a and the other parent was capital A, little a, we could represent the possible gametes that each parent could make and use a Punnett square to find the genotypes of the possible offspring. So we're going to be able to show segregation and fertilization. So this is what it looks like here. Okay, so this is parent one, this is the other parent. One is mom, one is dad. Okay, so mom has capital A, little a, and dad also has capital A, little a. Now remember, each parent can only pass on one of those alleles. Through segregation, they're going to separate it. One is going to get passed on. So remember, this kind of works like the old battleship, and what we're going to do is show where they intersect. Like this parent here is going to provide this one will provide this one. So we have capital A, capital A. Same thing here, capital A, but a little a here. Capital A, and it's always usually proper to put the capital A first, capital A, little a, and then little a, little a. So now we can look at this here. All the areas in brown here have the dominant allele. So that means all three of those will look the same. The phenotype will be the same, and so it'll be a purple flower here. And then there is one that is white, where we have the homozygous white, where we have the lowercase allele, the two recessive alleles together. That's the only case that we would have white. So if these are the parents, the chance of having purple offspring is 75%, 3 out of 4, and 25% white. But notice, in the case of the three purples, 2 of them are the same in that they're hybrid. These 2 are hybrid or heterozygous, which means in the next generation they could actually pass on the trait for whiteness. This one is homozygous purple, so it does not have the recessive allele, cannot pass it on. Okay. All right, so that's where we'll stop for today. In the next section, we'll look at dihybrid crosses and continue into the chapter from there.